So thank you, Jenny, and I'm the final talk for this session, and thank you to Dane and Daryl for putting this together along with the KI staff. And I'm not still quite sure if I count as an immunologist or an engineer, but either way, I and my new lab here at MIT have an interest in really understanding and then affecting recognition of immune receptors, and in particular, T-cell receptors. So in something that I probably don't need to motivate this room for, but T-cells, especially in the past several years, have had a really intense focus for a source of anti-tumor immunotherapy. And this has been done in all sorts of different ways, including trying to directly affect T-cells which are already presently in the body through either cytokines or through some vaccination strategies, or changing set points in terms of checkpoint blockade, things like that. Or by adding new T-cells, either with specificities which we dictate through engineered receptors, or through T-cells which we have some idea would have a good effect. And this has been done to really great effect. So we have survival plots from several clinical trials now, many, where the line plateaus and you see lasting benefit for some subset of patient. But as was mentioned earlier today, this line never always plateaus at somewhere that is not as high as we would want. And so a question which many people, including myself, have is what is causing this? So what's the difference between the surviving 50% here versus the non-surviving 50%? And is any of that difference attributed to what the immune response is seeing? So these are checkpoint inhibitors. So these are drugs which are going in order to reactivate or activate T-cells which are present in the tumor. And is there a difference, say, between what's being recognized in an effective response versus an ineffective response? So to understand this, so I spent my time before I came to MIT at Stanford really looking at some fundamental things about the T-cell receptor. And so the TCR is actually a fairly unique molecule as far as immune receptors go. Because first of all, it's restricted to binding to MHC, so essentially all alpha beta TCRs are going to be able to bind MHC molecules. These are also really amazingly low affinity interactions, so on the order of 1 to 100 micromolar is sufficient to activate a T cell. And in spite of that, it's also incredibly sensitive, so you can have just a handful of peptide MHC molecules able to activate fully activate the T cell. So this is a structure of this solved by my graduate mentor back when he was a postdoc. And you can see that the CDR3 loops are directly focused on the peptide. Everything else is focused on the MHC. And you look above, and we've really been able to begin to understand some rules for this interaction. So we know, for example, that if I take this one structure, again, here are CDR3s binding to the peptide. If I overlay many of them, you get what looks like this. So this looks like a bunch of spaghetti, but you can see that this isn't random. So there's still a polarity which is maintained, where the blue here, which is the beta chain, is over the C terminus of the peptide. The alpha chain tends to be over the N terminus. You have this general diagonal polarity, and you also have the focus of the interface. So right here, where the chains intersect, is focused on the peptide. So with this, we had some questions, one that's more of a practical question and one that's more of kind of a pure immunological question. So the practical question is, how can we find ligands for T-cell receptors? So as Jenny and Mark and others have talked about today, we're able to take T-cells which have some type of interesting effect. So whether they are expanded in a tumor or have an interesting activation phenotype or something like that. And we're able to isolate them, and with the advent of high throughput next generation sequencing, we're able to then go ahead and determine the sequence of their T-cell receptors. But taking this step, there's yet to be sort of a general way in order to do that. So to go from a known T-cell receptor to an unknown peptide MHC molecule. So there's no real robust experimental way to take the T-cell and really learn what it's recognizing. And the methods that are out there often had pretty low success rates and often have found relatively low affinity ligands. And for some things like some viruses which have fairly limited proteomes, there's ways to sort of brute force your way around that by able to just take overlapping peptides of the entire proteome and say we're going to find the epitope one way or the other. That's not as practical for some, either for autoimmunity or for cancer immunotherapy, or for some more complex organisms which have a very substantial proteome on their own, such as malaria or something like that. So that's the practical question of how can we do that. But that ties into kind of a bigger picture question, which is how cross-reactive are T cell receptors anyway? Because the immune response has a bit of a numbers problem to it. So that theoretically, there's about 10 to the 15 T cell receptors. As Mark mentioned, that's limited by just the number of T cells which are actually physically present in an animal, so it's orders of magnitude lower. And then the number of unique T cells is even lower than this. But the number of possible peptide MHCs, if you just sort of do the math out for what's possible for sitting in a peptide groove and then multiply that by the number of MHCs you have, ends up being a number which is as large or larger than, in fact, it's substantially larger than the number of unique T cells. So how does the immune system cope with this? How can you have a large universe of peptide MHCs which need to be recognized because we know that there isn't much of many holes in the immune repertoire, and we also know that you really don't want to leave holes in order to have something that could be exploited. But you also have to balance that with the ability to make sure that you have specificity in the immune response. Because if you don't want an immune response against a pathogen to be able to cross-react and affect you with autoimmunity. And so there's this famous figure from a theory paper from about 20 years ago where they did the math out, and they said that if there's one-to-one specificity, you would need a box of T cells that are 70 times the volume of this in order to cover this one mouse. So that's not the way that the immune system is able to work. But there must be some type of biophysical rules, structural rules, which are able to balance the need for cross-reactivity and the need for specificity. And so understanding this is also key to really understanding about what types of ligands we're able to target in something like cancer or for autoimmunity, and what is being recognized by a given T cell in a functional response that we'd be interested in. So we took, so this work was done in the Garcia lab at Stanford. We took a protein engineering approach to try to answer this in as unbiased and biophysically rigorous way that we could think of in order to do it. So we adapted yeast display to display peptide MHC molecules, which were attached to the surface of yeast, and then went ahead and diversified the molecules in order to get a library of a couple 100 million unique peptides. And so for this, the MHC molecule is invariant. And then selected with multimerized T cell receptors of interest in order to find peptide sequences, which we could then go ahead and analyze any way we wanted. So whether we wanted to look at the collection of sequences which were recognized by any given receptor, or activity, what is better or worse of an activator for a given TCR or T cell of interest, as well as structurally here. And so really the important thing here is that we make no preconceptions about what this peptide could be in our library. So we, in fact, do the best to make sure that anything possible that, so long as it can bind to an MHC, is covered here. And so the initial system that we used just to work out some of these cross-reactivity rules, as well as this proof of concept, was a mouse system, which I believe is as old as I am, which is the moth cytochrome C system. Sorry, front row. Older than I am. The moth cytochrome C system. So this is a murine TCR. So I'm going to talk mostly about a TCR called 2B4. It's a class 2 TCR binding to the MHC IE of K. Sorry for the mouse nomenclature word salad. But this is a system which has been as extensively studied a TCR as out there. So it's some of the first affinity measurements of recombinant TCR were made in the system. Daryl, as a postdoc in Mark's lab, really probed the sensitivity of the system. And in a collaboration between Mark's lab and Chris Garcia's lab, we also have a lot of structural information, as well as some peptide analogs to know exactly how this interaction occurs. So based on the structure, what we were able to do is take the MHC and reroute it into a smaller recombinant molecule where we preserve the peptide binding domain, so that's the alpha-1 and the beta-1 domain of the MHC, and link it to the peptide, omit the bottom domains here, which was more for protein expression expediency, although we found later on that this probably isn't necessarily strictly a necessary step. And we made this as a yeast construct. So peptide is linked to the MHC beta chain, and then the beta-1 is connected to the alpha-1, which is connected to the yeast. And so this construct, as something which we pretty heavily hacked together, was not functional at first, which is fairly typical for this system for different MHC alleles. But we were then able to go ahead and error-prone the construct and look for mutations which enabled recovery of staining, and we're using recombinant TCRs tetramerized in order to do this. And we found first through error-prone mutagenesis where we recovered staining a little bit for 2b4 and then more for 226, which is a higher affinity TCR. And then we combined that with some rationally designed mutations. So we saw that the error-prone was essentially solubilizing the bottom of the MHC peptide binding domain, which used to be part of a protein interface and was surface exposed once we were done with it. And so we also solubilized this patch of hydrophobic residues, and then we got really nice staining. We gained about a log. And importantly, none of these residues are affecting how the TCR would be able to recognize the MHC. So none of these are touching the peptide, or none of these are also on the helices, which would be able to interact with the TCR. So to the best of our ability, and we did some testing, this is still a very realistic representation of what could be recognized by a T cell receptor. Oh, I think I'm missing a slide. OK. So what we did next is we then took this construct and then randomized the peptide, except for the MHC binding domains, to ask the question, for this given MHC and this given T cell receptor, with no other preconceived notion of what the peptide would look like in order to bind, we went ahead and selected in order to see what motifs would allow binding. And when we initially started the project, we had it in our heads that we would get all sorts of very interesting different motifs, which potentially would move around the T cell receptor and give us a lot of things to study, structure-function relationships, and really probe cross-reactivity. It was going to be very interesting. But what we actually originally found is that this is the wild-type peptide where the interacting residues are A, Y, K, T. And we ended up with very, very similar peptides. So this K was absolutely conserved. This T was S or T, which is very, very conservative. AY went to AF. So when you give this T cell receptor and this MHC the ability to bind to anything at all, we found that the only answer that we came up with was essentially wild type. So we did all of that work to get the answer which we knew back already. So we weren't satisfied with this, so we went ahead and asked what else is in this library. And in order to do that, we turned to deep sequencing. So we did iterated rounds of selection, and I should say here that this selection and this strategy is a little different than protein engineering as it's normally done. Because with protein engineering, you're usually looking for the winners, or whatever molecule or collection of molecules have the property that's the best for whatever you're looking for, whether it's affinity, activity, or something like that. Here, we did the selections as sensitively as possible in each round using streptavidin-coated beads. Everything was done very quickly and in the cold, because we weren't interested in the winners so much as the players. We wanted to know every possible interactor, regardless of whether it was the best one or sort of at the bottom of our sensitivity limits. So we went ahead and did this. We did deep sequencing of each round of the library, and that allowed us to track enrichment of the sequences. And this was done in collaboration with Mark's lab here. And what we saw was, again, there was a few sequences that won out by the time the selections were over. So maybe 3 or 4 sequences made up 75% of all of the reads which we were able to see. But there were all sorts of other sequences here which were enriched. So here, gray is sequence 251 to 100 million, or as far as our reads would go at least, in terms of frequency. And the colors were the top 250. So you see that there was other stuff which was enriched and then competed out. So we were interested in what that is as well. And so what you're looking at now is the amino acid distribution of the 3 most important interacting residues for the T cell receptor, in this case, which was 3, 5, and 8. And you can see we do a good job of having broad coverage in our initial library. So we do a pretty even representation of all 20 amino acids. But by the time that you get enrichment, at the end here, again, only F, only K, with absolutely no tolerances. and here S or T. You'll also note that there were some other sequences that were unique motifs which were competed out. So this lysine was able to be a methionine or a serine, which are not traditionally thought of as obvious substitutions when you're thinking about this in terms of a protein interface. And similarly, S or T were able to become the hydrophobic residues, leucine and isoleucine, which again are not natural substitutions, or not substitutions that you would necessarily think of. And we did this for a couple of other receptors, which I'm not going to show you just for the sake of time. And what we found was, again, roughly the same pattern. So where the wild type motif, and it was still MCC, was preserved at the end, and then maybe some other things were allowed. And what those other things were varied sometimes, but they were always much less prevalent than the known wild type motif. So we asked a couple of questions about this, including affinity and activity. So I'm not showing you affinity data today, but I can tell you that this system has a sensitivity range of about 100 to 200 micromolar for interactions, which are incredibly weak interactions and probably rivaling the sensitivity of the T cell itself. We also did a sort of a broad look at activity of some of these peptides. So here I'm just plotting EC50s. Each of these is a different peptide. And in red is the wild type MCC peptide. So you can see that, again, we cover first, there's very few, if any, false positives here in terms of activity. Essentially, everything we tested was able to induce a T cell response. You'll also note that the wild type antigen is typically not the most potent antigen, which makes sense. So we were often able to find peptides with EC50s of 10 to 100-fold better than the wild type antigen. And unlike the B cell response, the T cell response has no ability to optimize itself during the course of activation. So there's really no reason that the antigen, which is the initial target of the response, is the most potent antigen able to induce that response. So that makes sense, but we saw that here too. The other thing we did, or another thing we did, was look at the interrelatedness between the sequences. And what we found was that all of the sequences were actually all three TCRs recognizing MCC peptide. If we do some clustering, again, this was in collaboration with Jake Glanville and Mark Davis's lab, we saw that if you basically do unsupervised clustering and ask for, basically take these and take a collection of naive library sequences, that essentially all of these sequences which were selected by one of these TCRs clustered with each other, and no sequences from the naive library clustered in. So all of these peptides, and this includes the peptides that didn't have obvious relationships that we competed away, ended up in this cluster. So in the Garcia lab, we had a fair bit of structural expertise. So I then went ahead and crystallized a few of these complexes with different peptides. And so here's a couple examples compared to MCC, which is the wild type peptide. And you can see, again, K to S, T to I, and then in this case, K to L, T to F for a 5cc7 recognizing peptide. And these are not conservative changes for the middle of a protein-protein interface. If I gave these on a test and asked, are these more similar or more different, the answer would be different. But when we overlaid all of these, what we saw was that in terms of binding footprint, these are essentially identical. So remember the spaghetti diagram, which I showed you on the first couple slides of the talk. This isn't that, right? This is almost an exact overlay, particularly for the alpha chain on the peptide, and pretty limited change in docking for the beta chain as well. So just to give one example for how the TCR is able to accommodate this, is that when we went and looked at how the CDR3 loop was interacting with the peptide, so what happened was that this loop was able to actually completely reorient itself. So here you can see that there's residues like S99 and W98 interacting with the peptide. You see the loop now, and this aspartic acid here. The loop actually folds in on itself and exposes an entirely new epitope, which is able to use to interact with the peptide in the groove. And so by repurposing residues which were in the CDR3 beta, it was able to make contacts which were compatible with this peptide. And the relatedness between the sequences and sort of why these clustered together, we're actually able to use our pool of deep sequencing in order to see this. We were actually able to take a sequence, so MCC, where the contact residues here are in blue, and then the peptide we call 2A, contact residues in orange, And we were able to actually walk one residue at a time for the contact epitope, where you can see that the changes can occur. So there's the relatedness that had these two get clustered together. And this is something where, when we have these two crystal structures, we can very easily rationalize this and really understand how the cross-reactivity is working. And when we have the sequences, we can rationalize this. But without that, if you had these just as 2 points, it would be very hard to come to the conclusion that these were going to be very naturally a cross-reactive pair. So, as a model here, the number we come up with for the amount of cross-reactivity is actually pretty close to in line with what people figured it had to be, which is each receptor has to be able to recognize something on the order of 50 to 100,000 unique peptides. But if you imagine that this is all peptide space, the cross-reactivity didn't occur sort of all throughout peptide space. You end up with what in this little blurb was called narrowly cross-reactive. So you are able to recognize sequences, a large number of sequences, but those sequences either obviously or sometimes non-obviously tend to be very heavily related to one another. So we then wanted to do a proof of principle here in order to see whether we can look at actually finding biological ligands. And so we collaborated with Kai Wucher-Fenning's lab at Dana-Farber in order to look at a TCR MHC system which was isolated from a patient with multiple sclerosis. And some cross-reactive peptides had been found for myelin basic protein, which is the endogenous ligand, and where from different pathogens which could be initiating events for autoimmune disease. And we wanted to know what other peptides can activate this TCR. There were some studies that were done where these peptides were found through no small amount of effort. And we wanted to know if we were able to find these, or even if we didn't know that myelin basic protein was the answer, would we be able to get that answer back? So we developed the MHC here required, HLA-DR15. Again, through some pretty analogous mutations, we had this functionally displayed on yeast. And then we also had a crystal structure from Kai's lab from a few years back, and we're able to then randomize and ask the same question. And something that I want to point out here is that if you actually do the math out for the number of unique either nucleotide or peptide sequences for diversifying all of this stretch, it ends up being a truly gigantic number, much, much, much larger than what could be covered in any yeast display library. So we are dramatically undersampling the number of possible peptides. So keep that in mind. But in spite of that, we did the experiment. And so here we know the most important residues are 2, 3, and 5. This time, there wasn't even anything else. So HF, HF. So we had the wild-type epitope and nothing but the wild-type epitope. A little bit more tolerance at the sort of the distal position for this interaction of P5. We, again, weren't satisfied with that. So we really went in to try to challenge that assumption. And we made a library where you couldn't have HF at 2, 3. We said we gave you all 19 amino acids here except H, all 19 except F here, and redid the selections. And what happened is essentially that the MHC outsmarted us. So in order to reestablish that epitope, the entire peptide register shifted over and repurposed an anchor residue here. So this F became the P3F. This was allowed to be an H, and the entire peptide moved over. So we got the same answer again. And when we tried to force the issue even more and not allow this to happen either, we got literally nothing out. So as far as we're able to say with our 100 micromolar affinity range, this is truly the only permitted motif in this system. So one thing is that we had the HF, but if you took any of these peptides, none of them are similar enough to MBP in order to be able to find MBP. So if you blasted our top hits, they're too different in other places to be able to find them. And with the library coverage, you would never expect MBP unless we contaminated our library to actually be able to show up, right? Like the chances of that are essentially 0. So what we did is we said, OK, we know for each position along the peptide what is or isn't allowed. Can we use the information from our library, even though it's a sparse sampling of what's allowed in nature, in order to use that to then search proteomic databases for peptides which exist in nature, which would follow this motif, and then say, OK, if these were in our library, we think that we'd be able to get those out. And then we went ahead and scored these hits. This was done, again, in collaboration with Kai's lab and with a postdoc in Chris's lab, and got a list of peptides. And so we had about 3,000 hits. Gratifyingly, the top hit, we did pull out the myelin basic protein. We also pulled out a whole list of other stuff, both environmental antigens and even other human antigens. These peptides were oftentimes quite different from MBP. So here in red is the sequence homology to MBP. So some of these only share maybe 3 or 4 amino acids in common with the wild type hit. But when we tested these for activity, we found that essentially all of them were able to induce T cell proliferation, and oftentimes at better peptide doses than what are often done for these types of initial pilot studies. And again, speaking to what Mark and Jeff talked about a little earlier about how is this working in terms of tolerance and T cell recognition. And I think that it really comes down to that there has to be enough cross-reactivity in the system built in that you couldn't ever exclude all self-antigens. Because if you had a system where no self-antigens were able to be recognized, you would by necessity then sort of get rid of anything able to recognize any other antigen. There just aren't enough epitopes out there. And also, just the idea of everything lining up in the thymus and very rigorously getting tested on its way out the door, it's how the textbook puts it, but probably isn't all that realistic either. So really, another vote for just the importance of peripheral tolerance. Because I'm pretty sure that essentially any T cell receptor of interest that you take out, even if it's isolated from something activated by flu, HIV, or anything else, we'd be able to find some cross-reactive human epitopes, which if you fed it to an antigen-presenting cell, would be able to turn that T cell on. And then gratifyingly, another group independently of us, I guess, read the paper and decided to put some of these peptides, which we found, to the test, and found that most of them were able to activate, or all of them were able to activate T cells, so they confirmed our result, and then found that a few of them were able to induce disease in a mouse model system. So in summary, we think that we found a mechanism for how T cell receptors are able to both be cross-reactive without being degenerate, and that cross-reactivity can often be found in sort of peptides which might look unrelated, but we can sort of rationally relate together. And we think that we're able to find biologically important ligands. So I've just started here a couple of months ago at MIT, but in the lab, we're interested in adapting some of these concepts to really get in to understand what's the difference between what's recognized or how it's recognized in a successful or an unsuccessful immune response, and whether we can sort of use these observations to go ahead and begin designing interventions which might be either more potent, more broadly applicable across patients. Can we get over the fact that we're all different in HLA type? And can we use this to make better engineered receptors? So can we do something to apply these types of mindsets to things like chimeric antigen receptors, engineered T cell receptors, and systems like that? So with that, this work was done at Stanford. I'd like to thank my graduate mentor and Mark and his lab were always a great resource. We also collaborated with the Wucherfenning lab quite a bit. And so that's the close of this session. So thank you. And we will, sorry, you can do that. OK.